Hi everyone, let's start with the next segment of microbiology, and that is virology. Now when I talk about virology, the first thing that we have to go ahead is general part, and if you get a good hang on this, trust me, half your life is sorted. General parts include the first thing that we all know, it's the smallest infectious particle, so much so that you can only see it on an electron microscope. You will never see me showing you pictures of light microscope over here. They're all electron microscopy photos. Next, they don't have ribosomes. That is one thing, but whenever I tell you something in general, I will always tell you an exception. So let me tell you an exception over here, and that is the arena virus. There is a virus called the arena virus, and that says that, like I said, all of them don't have a ribosome, but this has non-functional ribosomes. So what is this non-functional and where is it coming from? So this is something it gets when it enters our body. So it is the ribosome that it has stolen from us, okay? These are host ribosomes, and because it has stolen it from us, it is non-functional, right? So non-functional ribosomes in arenavirus, rest all of them don't have ribosomes. And lastly, they are obligate intracellular organisms. Do you remember, what does it mean? It's only going to grow inside a cell. So the good news is that I am not going to tell you any blood agar, culture media and all of those because they can't grow over there. They are needing cell lines. They'll need cells. Do you remember any bacteria also that was needing cell lines? So if you remember, which were the bacteria, which were the obligate intracellular organisms? We had the chlamydia and the rickettsia. We had chlamydia and rickettsia, which were also not having any of those agars. They were grown on cell lines, remember? Okay, these were the general properties. Now let's go on to the symmetry. The symmetry of whom am I talking about? I'm talking about the symmetry of the genome. That is, how is the DNA, RNA arranged? What all shapes can you see over here? You can see some random shapes, some nice kind of, not hexagonal exactly, but in that shape you can see icosahedral symmetry. All the DNA viruses are going to have the icosahedral symmetry. Next, you will see that RNA is looking something like a helical. So many of the RNA viruses have a helical symmetry. What are the two symmetries I'm talking about? One is icosahedral, one is helical. So now when I say that all the DNA viruses are going to have icosahedral symmetry, but there has to be an exception. And the exception to this is the pox virus, because remember, pox is complex. Okay, that is how I've learned it. Pox is complex. So the pox virus is going to have a complex symmetry. Otherwise, all the DNA viruses are going to be icosahedral. Talking about the helical. So usually what viruses come under helical are RNA. RNA can also have icosahedral symmetry, but there is a set of RNA which have helical symmetry. And I have learned that he is Mr. Fabulous. He is Mr. Fabulous means he stands for the very, very famous helical symmetry. And Mr. Fabulous stands for, let's begin. So this is going to be myxovirus, like orthomyxovirus, paramyxovirus, any myxovirus. Then with R, all of us know that this is going to be the rhabdovirus. Next, we have the filovirus. A we have as the arenavirus. And B we have as the bunya virus. So there are going to be many places where I will use the same mnemonic. Why I've made this mnemonic is कि हर जगह Mr. Fab use होगा. So you'll get to know that in a while. So quickly guys, let's revise. All the DNA viruses have icosahedral except pox virus, which is going to be the very, very simple. We write it as complex. We write it as complex. That has complex symmetry. And which are the RNA viruses with a helical symmetry? So myxo, rhabdo, philo, arena and bunya virus. Symmetries are done. Let's now go on to the rest of the structure. So till now I was telling you the symmetry of this. Now I'm going to tell you the entire structure in total. So let's begin from the outside. Of course, what you have on the outside is the envelope, which I'll talk about. And I can see some envelope proteins that are coming out of it. Can you see those projections? These projections that are coming out of it, these are the envelope proteins, and in many of the viruses, these are known as peplomers. Especially when you're talking about the coronavirus, very important. So remember, this is the envelope and you have a protein coming out which is known as the peplomers. What else? Ab andar dekhte hain. Over here, I see there is a capsid. The first thing that I can see over here is a capsid. And what is the capsid hiding? The capsid inside has some DNA or RNA, basically has the genome, has the nucleic acid. That could be a DNA virus, that could be an RNA virus. So some DNA or RNA is there. Ab in dono ko jod do. Combine these two. if I say that this is the nucleic acid and this is the capsid, together can I call it nucleocapsid? Kar sakte hai, right? So together they are known as the nucleocapsid. Simple. So one is the nucleic acid, one is the capsid, together nucleocapsid. What else? Not all, but some of the viruses. See, what was this? Nucleocapsid. What is on the outside? Envelope. You know this? Now can you see something in between the two? So sometimes in between the two, you have something known as tegument. Like I said, tegument is between the envelope on the outside and the nucleocapsid on the inside. So between these two layers, you have a tegument which may or may not be there. Let's move forward and now look at everything in detail. So I'm going to talk about the nucleocapsid separately. I'm going to talk about the envelope separately. Let me start off with this envelope. So what is this envelope made up of? We say it is made up of lipoprotein. Lipoprotein, okay. The lipid part has actually come from the host. We have contributed to the virus. And the protein part, the virus has on its own. So simple, the lipo wala part, L-I-P-O 1, 2, 3, 4, comes from HOST 1, 2, 3, 4. So remember, the lipo part we have contributed and the protein part the virus has contributed. Together it is made up of lipoprotein. So now you're going to say that so some of the viruses are going to have an envelope, whereas the others are going to be non-enveloped. So please note, the enveloped virus, they make it, you feel it has an envelope, it's going to be very strong, but its strength is its weakness. It makes it ether sensitive, alcohol sensitive, and heat labile. So heat can kill it, ether can kill it, alcohol can kill it. This means the enveloped viruses, can they be eliminated by all your hand sanitizers? Yes, because hand sanitizers contain alcohol based, right? So they can be eliminated by soap, by alcohol, by hand sanitizers. All of these can totally get rid of the enveloped viruses. Even for that matter, once by chance we end up eating this, koi fecal-oral route se, if we end up eating this, even our bile salts can destroy them. So that is good. Tell me, will enveloped viruses ever cause gastroenteritis in us? No. Why? Because our bile salts will kill them. So virus thought that, oh, I am getting an envelope and I'm going to be strong, but its strength became its weakness. That means that what are the other viruses which are non-enveloped? That is what I have to know. So there are a set of DNA and a set of RNA viruses, which is learnt as PAP and PARCH. What is PAP and PARCH? Parvovirus, adenovirus, papovavirus. Right now you don't know RNA DNA classification, which I'll tell you in 5 minutes. Abhi ke liye just learn PAP and PARCH don't have an envelope. So parvo, adeno, papova amongst the DNA family. And amongst RNA, we have picorna, polio wala. Polio goes into the alimentary canal. Astrovirus, this also causes diarrhea. Riovirus is rotavirus, that also causes diarrhea. Calcivirus also causes diarrhea. And hepatitis, I know hepatitis A and E also cause gastroenteritis. So I'm actually seeing my concept come true, that all these non-enveloped viruses are the ones which are associated with GI upset or gastroenteritis. I hope you've understood the concept. Why? Because they don't have an envelope, means they have my bile salts. Once I eat them, my bile salts or alcohol or all those spirits, they will not be able to destroy them. So over here, the envelope being there is a weakness. Means weakness that the virus will easily be killed by us. Envelope not being there is the strength. And who has the strength? PAP and PARCH. What will they cause? Gastroenteritis. So now, once you've done the envelope, now let's move on to what is the steps of viral replication that you have. Guys, before I start with viral replication, some 3-4 definitions we have to study. Okay? Firstly, virus ka replication and bacteria ka replication is not the same. Bacteria divides from 1 to 2, from 2 to 4. That is why we used to study the bacterial growth curve and all. Hai na? But for viruses, it's not happening like this. So, viruses are going to divide either in the nucleus, that is the DNA. So now let's begin with the next topic, and that is viral replication. Now, before I begin with this, let me make you familiar to a few definitions. First and foremost, what is a virion versus a viroid? Now, it's very difficult to understand this. First, I'll tell you the book definitions mentioned. Virion means it is a nucleic acid with a capsid, means it is a nucleic acid with a capsid. That is the virus particle formed. It is a virion. Whereas if only a naked RNA is there and there is no capsid around it. No, there is no capsid around it. That is a viroid. So how have I learned it? Virion has something on it. So there is going to be a capsid on the RNA or the nucleic acid. The capsid is on, like a covering, the capsid is on. And viroid is reminding me of devoid. So is it going to have a capsid? No, it is capsid devoid. This is the way of learning it. I hope you're finding it easy. Capsid is not there, devoid, so it is viroid. And capsid is on there, so it is virion. And what are prions? Prions are totally different. P for P. Prions are nothing but misfolded proteins. Prions are nothing but misfolded proteins that you have to know. So they are totally different from what we are talking about. Now let's go on to the viral replication. So if you feel viruses replicate like bacteria, not happening. Bacteria ka kya tha? Remember, bacterial growth curve. One bacteria dividing into two, two are dividing into four. All of that is not happening over here. There is no binary fission happening over here. This is going to be replicating in a different manner. Before I get to that, where are they dividing? So please remember, the site of replication for DNA viruses is the nucleus, because if I just go by my basic knowledge and if I draw a cell, koi bhi ek cell lo for example, and that cell has a nucleus, we all know that the DNA is going to be present in the nucleus and the RNA is going to be in the cytoplasm, isn't it? The RNA is going to be in the cytoplasm. So DNA viruses will always replicate in the nucleus except pox. You are seeing, ma'am, pox mein sab kuch complex hai. If you had remembered, all of them had icosahedral symmetry. All of them had icosahedral symmetry, all the DNA viruses, except pox. Similarly, all of them are going to divide in the nucleus. They are going to show nuclear, or not divide, I would say replicate. All of them replicate in the nucleus. Again, the exception to this is pox. So I say pox is very complex. Har baar exception mein hai. Amongst the RNA viruses, where do the RNA viruses have to replicate? They have to replicate in the cytoplasm, but which are the two deadly ones? One is orthomyxo. Orthomyxo matlab influenza. Influenza, to ma'am, now to we have experienced it, how influenza can, how these myxoviruses and all these can disrupt our life. So we know influenza is damn dangerous, and retrovirus I know as HIV. These are the ones which are exceptions. They do not replicate in the cytoplasm. So repeating once again, all the DNA viruses replicate in the nucleus. All RNA viruses replicate in the cytoplasm. What are the exceptions? DNA virus ka exception is most of the times pox virus, because it is very complex. And RNA viruses mein I have two real deadly ones, HIV and influenza, orthomyxovirus. Now let's start with the very, very famous viral replication. These are the steps. No one can learn them in an order. So I've got you a cute little kiddish mnemonic. Appu the elephant is sitting on a bar stool. So, Appu on a bar stool is going to be your 6 to 7 steps of the viral replication that you have. So, let's start one by one. What do you see? If this is the cell, what does the virus have to do first? The virus first has to come and A for Appu ka A for attachment. So we are going to go exactly in that order. Everywhere I will write Appu on a bar stool, because A is going to be attachment or absorption, and you might feel that this is just common sense. Virus has to attach, but do you know this is the most specific step? The virus coming and attaching is the most specific step. Very, very, very important. And please note that this attachment that is occurring is because of some receptors. If this is a virus and this is a host, there are some receptors. I'll give you some examples. Everyone's heard of the famous HIV virus. What does it have? It has GP120 and GP41. That is what it uses. So God has given it 120 and 41. That is how it will come and attach to my cells. Okay, coming to the next. Everyone has heard of influenza virus, like I'm saying. The deadly ones I'm talking about. So God has given it H and N proteins. H and N, especially H is going to help in coming and attaching. Now let me ask you, हमारे पास क्या receptors हैं? So let's give another example, which is not an example that you have to know. SARS-CoV-2. SARS-CoV-2, can you say, comes and attaches to which receptor of our body? Which receptor in the humans? We know it is the ACE2 receptors, yes? The ACE2 receptors in the body is what it comes and attaches to. ACE2 receptors are there in the lungs. That is why coronavirus comes and attacks the lungs, right? So either ways, the virus has also been given some special mechanisms. We unfortunately also have some receptors which will receive the virus. So that is how the attachment will occur. So Appu ka attachment is done. Now it's time for the virus to enter. So what is it? Again, we do the same mnemonic. Appu on a bar stool. So A was for attachment. P is going to be for coming inside the cell. Penetration, also known as the next P, pexia. So remember, penetration or viropexia means the same. So it's not that easy that virus just comes inside my body. That's not what's going to happen. Please note that the virus is coming in by many methods. What are the methods? Number one, I just eat up the virus. What is that eating up known as, ma'am? The very famous phagocytosis, which we are famous for doing. So either our macrophages have just eaten it up. Second, the virus says, nahi, aise nahi, we are going to have a contract, mutual contract. Virus ka membrane, my membrane, they are going to fuse. So the second method is going to be membrane fusion, and you will study eventually in the HIV chapter that membrane fusion is a method by which HIV enters the body. And third is, do you all remember a virus? I'll draw the shape. Let me see how many of you remember this. ये कौन सा वाला virus था? Ma'am, the tadpole. This was a bacteriophage. Bacteriophage is also a DNA virus, na? So imagine if a bacteriophage has to enter into our body, bacteriophage doesn't enter on pura tadpole doesn't enter into the cell. No. You know this DNA virus, bacteriophage, it kind of spits. It says, I am just going to send, I'll show you that this is the tadpole. So this bacteriophage says, attachment to theek hai, I will follow. But for penetration, I don't have so much of time. Pura tadpole is not gonna come inside. It says, I'm just gonna, can you see it has spit out its DNA? I'm using a very crude word so that you remember that it's like spitting out the DNA, that I'm spitting inside your cell and I'm gonna go away. I am not gonna come. I don't have so much of time. So the third method is that, now let's put it in technical terms rather than saying spitting out, injection of the genome or the DNA by the bacteriophage. That is the third method. So what are the three ways by which the virus will finally penetrate into our cells? Number one, by phagocytosis. Number two is going to be by membrane fusion. And number three is going to be injection of the DNA only. So the virus doesn't come, only its DNA comes. Attachment ho gaya, penetration of pexia ho gaya. Now what do we move on to next? The next is going to be uncoating, which I'll explain with the same diagram earlier. So have a look at this. You'll say, ma'am, if the entire virus had come inside, I am not interested in the entire virus. I am only interested in its DNA or RNA. DNA, RNA uthao and start replicating. I want to get rid of all those envelope and tegument and capsid, remove all the layers. And that is what is uncoating. What is uncoating, guys? Uncoating is all the capsule or envelopes, whatever enzymes we have, our lysosomes, they go and say break it off. So what a capsule, lysosome, jo bhi hai, I'm interested in that DNA. So my primary concern is get rid of all these coverings. How will I get rid of these coverings? By the lysozymes. Finally, please remember the nucleic acid is released. I was interested in this nucleic acid. This is released. But what do you think, and in which virus will this step never be seen? Pehle to dekho. In this particular situation that I'm talking about, you will say, yes, ma'am, I can see all these layers have been removed. And now you've got only and only the DNA that you were interested in. Uncoating completed. But what if I'm talking about a bacteriophage? Bacteriophage, though, from beginning only was very clear. I don't want to send things that you're going to kill eventually. You need the DNA. I am sending the DNA. Do you do uncoating in this kind of an entry? No. Can I say this step is absent in bacteriophages? This step is absent. Imagine. So in Appu on a bar stool, if they say that which is the step of this replication which bacteriophages will skip, you will say there is no uncoating. So you will ultimately realize that if other cycles were taking 30 minutes or maybe some 1 hour, bacteriophage will take half the time. Ek step khatam ho gaya na. So bacteriophage ka this replication is going to be shorter, which I'll tell you in a while. So everyone's understood. Everyone has uncoating to release the DNA, RNA, but bacteriophage doesn't. Okay, so Appu is done. Now we come to the bar stool, and that is what it is. Now you have got your, everything is done. You got your main thing, the DNA or the RNA. Now start making new virus particles. Now start doing biosynthesis, start making new virions. You start making, once again, you such a DNA, hey, let me make a capsid, let me make a tegument, let me make an envelope. You're making new things. So biosynthesis kara. Once you make new things, you start arranging them. Assembly karo. Assembly and maturation. You do assembly and maturation. And finally, once the virus is ready, you will say it's time for you to get released. How are you going to release it is also important. Please note, number one is this cell says, I am going to die out. I have so many viruses, I'm going to burst open, lysis, and all the viruses are free. So that is lysis. Second, the cell says, why should I die? Why should I sacrifice my life? ऐसे spit out. Again, virus को बस ऐसे throw the virus out of the system. I am not going to sacrifice. So that is budding out through the host membrane. So two things. Either this cell will sacrifice and lice open up, or this cell will say, why should I sacrifice my life? Just chuck out the virus out of my cell. That's it. So this is finally the completion of Appu on a bar stool. Let's do the quick revision. Appu, A for attachment. Attachment, if you remember, is the most specific step of this cycle. P and P refers to viropexia or penetration. That's the second step that is important. The third one is going to be uncoating. That is the step which is absent in bacteriophages. Now coming to bar stool. So B is going to be make new viruses. That is going to be biosynthesis. After you've made the new viruses, assemble them, let them mature. So assembly and maturation. And finally, you're going to release those viruses, and that is the last step. So that is Appu on a bar stool that is available. Guys, what do you think is an eclipse phase? You will say, ma'am, from the time that this virus entered, dekho, from the time that this virus entered to the time when you made the first viral particle, from entry to synthesis. From entry to synthesis is the eclipse phase. So remember, between the entry and the first virus formation, that is eclipse phase. Now you will understand that for animal viruses, it takes almost 30 hours, 15 to 30 hours. But bacteriophage mein kam time lagta. How much? Just 15-30 minutes. And I've told you this concept. Why? Because the uncoating step was missing. If one step is absent in Appu on a bar stool, of course it's going to take less time, but itna kam time. So animal viruses are going to replicate in 15 to 30 hours. Bacteriophage, it's only within half an hour. That is 15 to 30 minutes. And that is what you have to know for the replication. Moving on to the next part. And now we start with the classification of the viruses. Please note, we can have either a set of DNA virus, that is one family, or they can have RNA. So basically we realize that only one of the two can be there. Either DNA viruses or RNA viruses, both can't be present in the same virus. So you will have to decide, does it have a DNA or does it have an RNA? I'll show you the RNA virus ka list, and please don't run away or shut off the video because you might feel, arey, to nahi hone wala. Ma'am had said bacteriology is difficult. Now she's shown us so many viruses under virology also. So we have picorna, which is polio wala, paramyxo, calci, toga, orthomyxo, bunya, flavi, arena, corona, rio, rhabdo, retro, philo. Okay, याद करना है? नहीं, we don't want to learn. कोई छोटा list है? Yeah, RNA viruses are very difficult to learn, so I give up on them. I tell you, DNA viruses याद कर लो. This is a shorter list. So whatever you will learn here becomes DNA virus, and after this, anything that the examiner asks in viruses becomes RNA virus. ठीक है? So what are the DNA viruses? And I've got you a happy mnemonic. So I've made you happy that छोटा वाला list याद रखना है. Right? We are gonna do the happy mnemonic here. What is happy? H for hepadna. Hepadna means hepatitis, the only hepatitis virus which is DNA. Hepatitis B, this means after this, all hepatitis A, C, D, E, all hepatitis viruses will go under RNA. The only hepatitis is going to be hepatitis B, which is the first H. The next H is going to be the herpes viridae family. You have HH, I'm not going to talk about it in detail. You have human herpes virus 1, 2, 3, 4, 5, 6, 7, ऐसे करके. Some of the examples I'm showing you over here, like herpes simplex virus, herpes zoster virus. So obviously all of them will be herpes. Herpes simplex virus, herpes zoster virus, human herpes virus. तो सब में herpes आ रहा है. EBV and CMV, Epstein-Barr virus and cytomegalovirus. They are also human herpes virus 4 and 5 respectively. EBV is human herpes virus 4 and CMV is human herpes virus 5. So ultimately, right, I'm only making you familiar. These are all separate chapters, okay? So we're going to study all of these in detail. So HSV, HZV and EBV, CMV, HHV, all of these come under it. A for adenoviridae. This is my favorite family because there is adenoviridae mein adenovirus. Life is simple. Now we come to 1P, which all of us already know, bacteriophage. We've done that tadpole-shaped bacteriophage so many times, yes? So this is the bacteriophage, which is a DNA virus. What else? Then we have the very famous parvovirus. This is also my favorite because parvoviridae family has parvovirus. See, simple life. Now we come to the pox virus. Pox has all those small pox, yes, cow pox, not chicken pox. Please don't put chicken pox over here because chicken pox is herpes zoster, right? Not chicken pox. So ये बहुत common confusion है. Pox virus is small pox, cow pox, molluscum. So this includes Vaccinia, variola and molluscum. Repeating, Vaccinia, variola and molluscum, the smallpox, cowpox and molluscum, they come under pox viridae. Finally, coming to the last one, that is papova. Papova ka thoda break karo. What are rhyming words I can think of? Polyoma sounds like this. Papilloma sounds like this. So please remember, we have the polyoma virus. We have human papilloma virus. तो papova में polyoma, papilloma सारे rhyming words आएंगे. We have BK virus, JC virus. Now we have another very important one, that is SV40. तो ये जितने short form ones are there, and papilloma, polyoma, and सारे rhyming things are coming. So papilloma, polyoma, BK, JC, SV, all these short forms, that is papova viridae. That completes our happy mnemonic. A quick review: hepadna, herpes, adeno. Then there is parvo, bacteriophage, papova, and pox. That makes us happy. That is the DNA viruses. Now let's talk about the different types of sizes of these viruses. Now we know all their names, so now life will become easy. How do I measure the size of this? There are two I told you, they are very, very small particles. You can't see them on a light microscope. Which microscope are you going to see them on? You will need an electron microscope. Apart from that, there are two ultra techniques that are used. One is known as ultrafiltration. The other is known as ultracentrifugation. So these three techniques are used for knowing the size of a virus. And out of those, please note the largest one I told you, pox, was always complex. Everything was a little different about it. It's the largest virus, measuring 300 nanometers. And the smallest is going to be parvovirus, which measures 20 nanometers. So the range of the size of viruses is going to be from 20 nanometers to 300 nanometers. Repeating, 20 nanometer wala is parvovirus and 300 is going to be pox virus. Now the next property that we go on to is going to be the shapes of these viruses. They have some amazing different shapes. What I'll do, there are some pictures for these that you have to know. We'll first do some pictures and then we'll come back and see if there's anything extra that is left. The first one that I see over here, see, all of them are black and white photos because which microscopy? Electron microscopy. The first one that we have over here is going to look like a bullet, and bullet virus is known as a rabies virus. So a bullet-shaped virus is rabies virus. I have learned that bullet is also going to kill and rabies is also going to kill. Hai na? So bullet is rabies. The next virus is the one that has changed or ruined 3 years of everyone's life, and that virus is the one which has these papillomas. So when we are talking about papillomas, you are of course reminded of the very famous coronavirus, because the meaning of the word corona is that it's going to have a crown around it, right? So coronavirus is going to be a crown-shaped virus. Moving on to the next, we have a virus which is looking like a box. So you call it a box shape, or you can also call it a brick-shaped virus. And inside that box, I can see a dumbbell-shaped DNA core. There is a dumbbell DNA core that is going to be present inside it. So a box-shaped virus with a dumbbell DNA core, have a look at this. A box or a brick with a dumbbell DNA, and that has to be the heaviest one, sounding quite heavy. So that is the pox virus. So now you know pox virus size, maybe it's 300 nanometers, the biggest, and heavy bhi hai because it has a brick and a dumbbell inside it. Now the virus which is looking like filament. So they ask you which is the filamentous. In fact, if you want to imagine it, this is actually looking like a bowl of spaghetti. So what is the virus that looks like a bowl of spaghetti? Remember, it is Ebola virus. Very simple to learn, that Ebola virus is going to look like a bowl of spaghetti. That is my way of learning it. Spaghetti pasta, right? Moving on to the next. This is the easiest one because it's looking like a star. Anything looking like a star is going to be an astrovirus. So it is a star-shaped virus. Moving on to the next, the one that is looking like a rotating wheel. It's looking like a rotating wheel, so that has to be rotating, rota. This is going to be rotavirus, and if you ask me what family does rotavirus belong to, it belongs to the Rheoviridae family. It belongs to the Rheoviridae family. So remember, rotavirus for Rheoviridae for rotating wheel. Moving on to the next, the one that looks like a spaceship, as if an alien has come in a spaceship. The one that is looking like a space vehicle, and like I said, space vehicle. Who will come in a space vehicle? Alien will come. So this is said to be the A for A, adenovirus. Some adenovirus alien has come, so this is the shape of adenovirus, like a space vehicle. The one that looks like a cigarette. Okay, you can see these rods which are present, rod shaped. So cigarette has tobacco. Cigarette has tobacco, so this is known as TMV, tobacco mosaic virus. Tobacco mosaic virus is going to be a rod-shaped virus. So I've learned tobacco is in cigarette. It looks like a rod. Quick revision of photos and then quick revision of the text. The one that is going to kill is a bullet, rabies is going to kill. The one that looks like a crown with peplomers, coronavirus. The one that looks like a brick with a dumbbell, that is going to be pox virus. The one that is a bowl of spaghetti is going to be Ebola virus. The one that looks like a rotating wheel is rotavirus. The one that's looking like a star is, of course, astro. The one coming in a space vehicle, alien, that is adenovirus. And finally, looking like a rod or a cigarette with tobacco, tobacco mosaic virus. I think you have done this. Now, if I ask, I'm not going to write. Now you know. So, rabies ka bullet shaped. Pox is a box with a dumbbell DNA inside it. Ebola is going to look like a bowl of spaghetti. Tobacco mosaic virus will look like a cigarette. Adeno was alien, so this is going to be the space vehicle virus. Corona, of course, is going to be the crown with the peplomers. Rota is going to be the rotating wheel. And astro is going to be star. Ho gaya revision? The shapes are done. Let's move on to the next. We have some rules. What is this, ma'am? Why have you correlated it with Bollywood? Now, didn't I tell you happy viruses, DNA viruses? So now come to some rules. All the DNA viruses are what? All the DNA viruses have a double-stranded DNA except parvovirus. Except parvovirus, and that parvovirus is having, so paro, that is how I've related it to. So paro, you are realizing that why is this paro crying so much? Because she is left alone in life. Na? She did not get her husband, she did not even get Devdas. So she was left alone in life. That is why she's sulking, that is why she's crying. She's left alone. She is not double-stranded. She is the only single-stranded DNA virus. Not only that, if you remember, Paro is also crying because she is the smallest virus. That is what I told you. Parvovirus was that 20 nanometers wala. So repeating, all the DNA viruses are double-stranded except for parvovirus because Paro is left alone. So she is single-stranded and also she is the smallest and hence she is crying. Right? The continuation of this mnemonic will happen. She's crying, she'll become red. So this is gonna result in slapped cheek appearance. All that continuation of this movie and mnemonic will happen under the parvovirus family. But for the time being, DNA viruses ka exception is done. Now let's move on to the... So what are the exceptions for the RNA viruses? Please note that all the RNA viruses are going to have, first I'll tell you a basic rule about them. All of them have only one copy, okay? All of them have only one copy of what kind of RNA? Of single-stranded RNA. And please note that RNA is unsegmented. What all did I mention over here? All of them have only one copy. Let us draw. This is that one copy, only one copy is there. And I have not drawn it like this. I have not made it double-stranded. I am saying it is alone. It is a one copy, one strand. One copy, one strand. And one piece. I have not made it into segments. I have not made it like this into multiple segments. No, I have learned that all the RNA viruses are one copy, one strand, one piece. Okay? One copy, one strand, one piece. That is all the RNA viruses. Now let's come to the world of exceptions. So please note which is the virus which has everything same, but instead of one copy, it has two copies of. So what will it have? It will have two copies of single-stranded unsegmented RNA. That is how we'll do. So only thing that has changed is it has two copies like this, and that is known as the retrovirus. How I have learnt it is, retrovirus matlab HIV na? For how I have learnt it is the very famous Red2 virus. Red2 virus will tell me that okay, this has two copies. Simple. Okay, all of them are going to be single-stranded. I will need one exception over here also. So which is the one which is going to be double-stranded RNA? So you will say that, ma'am, think of something that causes diarrhea. Diarrhea wala jo RNA hai, that is going to be double-stranded. And the famous diarrhea wala RNA is going to be the famous, guys, rotavirus. Rota or Rio, they belong to the same family. Matlab, rotavirus is Rioviridae family. So the rotavirus is going to, we know na, rotavirus causes diarrhea in children. So the diarrhea wala virus is going to be double-stranded RNA. And finally, all of them are single piece. But which of them are going to be segmented like this? How are they going to be segmented? So segmented matlab instead of one piece, they are all bikhra hua. Who is going to bikhro? The person who will drink. What has he gotten drunk on? So please remember, he has started drinking bira. He has started drinking bira. So bira refers to B for bunya virus, I for influenza virus, R for again the Rio or the rota virus that I was talking about, and A for arena virus. So repeating, bunya, influenza, रियो अरीना. इन्फ्लुएंजा मतलब कौन सा वाला इन्फ्लुएंजा? मिक्सोवायरस, ऑर्थोमिक्सोवायरस, पैरामिक्सोवायरस. Remember, it is going to be the orthomyxovirus. So, bira, bora is what you have to remember. And someone who drinks bira, bira is an alcoholic beverage, which means that person is going to break into pieces. How many pieces is this virus going to break in? So, remember, the B looks like a 3. B is going to look like a 3. So, how many segments does it have? Now, you need to know how many segments am I talking about. In Bunya virus, there are 3 segments. In influenza, influenza looks like I looks like 8. So, in influenza, there are 8 segments. 3 plus 8 karo 11. So in Rio virus or rotavirus, there are going to be 11 segments. 1 plus 1, 2. Arena virus has 2 segments. Let's do a recap, guys. What is this bira mnemonic? Bira, if you drink bira alcohol, you are going to break into pieces. Basically, you're going to fall here and there after drinking alcohol. So the RNA has also fallen into pieces. B looks like a 3, so it has 3 segments. So B ka I made a 3. I ka I have made an 8, so 8 segments. Then I've done 3 plus 8, that makes it 11 for rota, and 1 plus 1, that makes it 2 for arena. That's your final list of the RNA viruses and exception. A quick revision: all the DNA viruses were double-stranded except Paro or parvovirus, who was left alone single-stranded. All the RNA viruses have one copy of one strand and one piece. One copy, one strand, one piece. But which is the one that has 2 copies? Red2 virus, which is double-stranded, diarrhea wala rotavirus, and which is going to be segmented, all the beta bora viruses. Let's move forward. All of them are usually positive sense RNA. The story is not over. All of them are positive sense RNA. Matlab, positive sense RNA is the one that is directly going to make the proteins. Whereas we have something called Mr. Fab, which are negative sense RNAs. Now, what is negative sense RNA? Negative sense, you will say, ma'am, this so cannot make the proteins. You will have to convert a negative sense RNA into a positive sense RNA, and then it will make a protein. And how do I convert that? By RNA dependent RNA polymerase, RDRP. What did I say? By RNA dependent RNA polymerase. RDRP is the one that is going to be making it a positive. So simple. If it is a positive sense RNA, it will directly make protein, and that is what all the RNAs are. But this negative sense wala is first going to be converted into positive sense by RNA dependent RNA polymerase, and then it's going to make a protein. So remember, Mr. Fabulous, when someone is becoming fabulous looking, very, very good looking, to Mr. India mein Mr. Fabulous bana. On his own, he has not become. He has, initially, he was not that good looking. Then a lot of people worked on him, makeup, hair, all of that happened. Then he became famous. Okay, so Mr. Fabulous is negative sense. And you've done this Mr. Fabulous earlier. You know this belongs to Myxovirus family. Then R is for Rhabdoviridae family, F for Filovirus, A for Arena virus, and B for Bunya virus. All these RNA viruses, where else did you do Mr. Fab? So I'm saying na, he was Mr. Fab. All of them had helical symmetry. So now I know that he, that is Mr. Fab, not only has helical symmetry, but is also going to be negative sense. It is also going to be negative sense RNA. Very important. I hope Mr. Fab mnemonic ka version is clear. Helical symmetry and negative sense. Let's move forward. Now, I told you, if they ask you the size of the virus, if I would have asked you which is the largest, you said pox. And if I asked you what was the smallest, you said, ma'am, that was the parvo, chota wala, crying wala virus. But now I'm asking you the size of the genome, the DNA and the RNA. So don't get confused in the paper. What have they asked me? Size of the genome or size of the virus? Because size of the genome, which is largest, has to be HIV. Come on, that has to be the retrovirus, largest genome. Smallest genome, there's always a confusion, I'll settle that. It is actually hepatitis D. And if hepatitis D is not there in the options, then you say hepatitis B. So previously a question came, they asked you which is the smallest genome. Hepatitis D was not there in the options, so students selected hepatitis B. But after that, many students do question answer only. Na? So they started doing PYQs and they started learning smallest genome is hepatitis B. No, that is only if the hepatitis D is not mentioned in your options. Hepatitis D is the smallest genome, followed by hepatitis B. Largest, retro, smallest, hepatitis D. Done. Now finally we come down to the next topic. All your exceptions are done. Now we move on to how are you going to cultivate the virus. For virus cultivation, did I tell you they are obligate intracellular organisms, which means you cannot cultivate them inside any blood agar, chocolate agar. You need cells. So cells के लिए I got an animal. Animal has cells. So you're going to inoculate it into the cells. Who? Which animal? Brain of the suckling mice. Now let me tell you, there are two viruses which are very important. There is a Coxsackie A virus and B virus, and also some rare ones, but arbovirus. Arbovirus can also be inoculated in the suckling mice. But my primary concern will always be Coxsackie. So this is what is the question they will ask you when I teach you Coxsackie A and B. So imagine if you inject Coxsackie A into the brain of a suckling mice or Coxsackie B, तो पता कैसे लगेगा whether A was injected or B was injected? So one of them is going to result in flaccid paralysis in the mouse. In these mice, there is going to be A resulting in flaccid paralysis and B is going to result in spastic paralysis. B is going to result in spastic paralysis. So repeating once again, A will result in flaccid, B will result in spastic. How do I learn? Put it in alphabetical order. A comes before, B comes after, F comes before, S comes after. So alphabetical mein lagao, A with flaccid, B with spastic. So Coxsackie can be grown in brain of suckling mice, arbovirus can be detected over here. Animal inoculation done. But do you think that's easy to find suckling mice and then do the identification of a bacteria? No. Then we said, why don't we keep some embryonated eggs in our laboratory? Because embryonated eggs to we will find. And inject in an embryonated egg and the virus is going to grow. So before I tell you all of this and what are these small beautiful pictures that I've got here, embryonated egg hota kaisa hai? What are the parts that I have? I can see there is an amniotic cavity, there is a chorioallantoic membrane, there is an allantoic cavity, there is a yolk sac. These are the parts of an embryonated egg. Na? And what have I got? I have got a car over here and I've got these two boys who are best friends. Their pose is that of a best friend. So remember, all the boys are very, very, you must have seen, all, not just boys, even girls, but over here I've got a boys ka mnemonic because it's fitting well. All the boys are very possessive about their boy gang and their yari with their boy gang. They are very, very protective. They are very, very protective. Me and my boys is like a big thing amongst the boys, right? So boy gang, yari gang, they're very possessive about it. That is one mnemonic. Another things that boys tend to obsess over are cars. So if you remember boys and their possession, obsession with their yari, protectiveness with their yari and their protectiveness with their cars, I think this mnemonic is going to be simple. Now when I talk about the embryonated egg inoculation, tell me all of them are going to be very, very protective. All of them are very, very protective about their yari. Means what? That in the... So what does the all over here stands for? All stands for allantoic cavity. And what is the yari? So Y stands for the famous yellow fever. The R is going to stand for rabies and I shall stand for influenza. Now there's a catch over here. Influenza, you will also study it in the amniotic membrane and you will also study it in the allantoic cavity. The amniotic cavity and allantoic cavity. But if you want to grow influenza, you will grow it in the amniotic cavity. If you want to make the vaccine of influenza, you will do it in the allantoic cavity. That is, I said, na, vaccine matlab kya? Vaccine matlab protection. So that is what I told you. Influenza ka protection is in the allantoic cavity. Now your mnemonic is complete. All the boys are very protective about the yari. So all stand for allantoic cavity. Protective means protection for influenza. Vaccine for influenza is grown over here. Y for yellow fever, R for rabies. Sorted. In amniotic cavity, you grow influenza. That is also done. Yolk sac. Since kids, yolk sac matlab chota sa yolk sac. Since kids, boys are very, very obsessive over their cars. What is cars? So yolk sac mein cars. Virus mein A for arbovirus. But C and R are chlamydia and rickettsia. Because if you remember, these were the two bacteria that I told you were obligate intracellular. You could not grow them on blood chocolate agar. You had to grow them on cell lines and embryonated eggs. So exactly, chlamydia, arbovirus and rickettsia, these are going to be grown in the car. Talking about chorioallantoic membrane, that is CAM, this is VVHH. What is VVHH? It is Vaccinia, Variola, HSV1, HSV2. What did I say? Vaccinia, Variola, HSV1 and HSV2. So VVHH. Let's do a repeat of this entire mnemonic. Repeating once again. Let's do it over here. If I say chorioallantoic membrane, CAM, so you will say, ma'am, VVHH. That is Vaccinia, Variola, HSV1, HSV2. Perfect. If I say amniotic cavity, you are going to grow influenza in it. But if I say allantoic cavity, all the boys, allantoic cavity, are going to have yellow virus, then it is going to have rabies virus, then influenza ka kya? Influenza ka vaccine is going to be here. And finally, we come to the yolk sac. Since yolk sac boys are very obsessed with cars, so chlamydia, rickettsia, arbovirus. All of this makes up your embryonated. I hope it's becoming interesting because now we were doing virus cultivation. First, you could use an animal inoculation. Second, you could use an embryonated egg. But all of these require a lot of logistics. So what we actually do is a tissue culture using cell lines we have prepared. Like in bacteria, we had prepared blood agar, chocolate agar, and kept lots of plates in your lab. For viruses, you have made some cell lines and kept them in your lab. So what are these cell lines? Primary, secondary, continuous. Primary cell lines means how many times can the virus multiply in it? 5 to 10 divisions. Secondary में थोड़ा ज़्यादा होगा, so almost you're going to add one more zero in front of it, so 10 to 50. So remember 5 to 10 and secondary में 10 to 50. But if I ask you what is it with continuous cell lines, you will say unlimited. Actually, it's not unlimited, but yeah. For practical purposes, we say continuous and unlimited divisions can be allowed over here. So repeating, for primary cell line 5 to 10 divisions, for secondary cell line 10 to 50, and for continuous cell line there are continuous divisions. How am I going to learn this? देखो, primary school मतलब primary छोटा बच्चा, तो all the things that have embryo, amniotic, these kind of words in it, या फिर humans का precursors कौन थे? Monkeys, rhesus, anything that has primitive words in it will come over here. I'll give you an example. So look at this. If they say rhesus kidney cell line, rhesus were our ancestors, monkeys, तो वो primary में आएगा. Human amniotic cell line, amniotic cavity from what we have come, bachas, so that is also coming here. Chick embryo fibroblast cell line. Anything that has embryo, amniotic, primitive primates का नाम, that is going to come under primary cell line. Now let's make this bacha a little big. Instead of human amniotic cell line, if I now say human fibroblast cell line, you'll say, ma'am, isme there is no word called amniotic or rhesus or nothing. Fibroblast matlab bada person ho gaya. So now I will put it under the bada person, that has come under secondary cell line. And this is particularly used for the growth of cytomegalovirus, a very famous previous year question. Which is the cell line used for the growth of cytomegalovirus? It is human fibroblast cell line. So be cautious. Human amniotic cell line पूछा तो you put it under primary. Human fibroblast cell line पूछा, you put it under secondary. Other than that, M and W. So remember, MRC5 and WI38 are also going to come under secondary cell lines. Now let's move on to continuous. After this, whatever they shall write in the exam, be it HeLa, Hep2, KB, McCoy, Viro, Detroit, VHK, Chan, CILK, कुछ भी लिखा हो in the paper, all of them are cell lines. So you must be wondering, ma'am, where are these cell lines coming from? I think you can guess. This rhesus has been taken from a monkey. Human amniotic has been taken from our amniotic cavity. The chick embryo cell line has been taken from the embryo. Human fibroblast cell line has been taken from our fibroblasts. Viro cell line has been taken. Viro is an African monkey. Viro is an African monkey. So this has been taken from African monkey. Basically, the concept is that all your cells have to be taken from someplace and you can make many preparations out of it. Okay with everyone? So all your cell lines are done. What is the next topic that you have to do? So finally, before I proceed, a quick revision of all this. I want to grow the viruses. Number one option, I can grow it inside animals. Second, I can do an embryonated culture, which I did all those allantoic, amniotic, sab kuch. And third, I did tissue culture using a lot of cell lines, primary, secondary, and continuous. Now I come down to what are the inclusion bodies that the virus can make, and these are known as CPE. What is CPE? Cytopathic effect. CPE is known as cytopathic effect. So you must have seen that when bacteria I was talking about, I could say, oh, look at this, bacteria is looking like cocci, like bacilli, like lens shape, grape of cluster, because you could see them under a light microscope. Na? I cannot see viruses under a light microscope. Then what will I see? This is the effect of a virus. This is the effect of a virus on a cell that it has created. So indirect route, a pathologist or microbiologist क्या करेंगे? They have a microscope. They say either I see the organism. If I cannot see the organism, I will catch it from a different way. I will see what change it has caused in the cell. So what cell change has happened, that is known as cyto for cell, pathic for pathology. What is the pathology in the cell that it has created? So remember, some changes it can make in the cytoplasm, some changes it can make in the nucleus. Let me ask you, what are the changes in the cytoplasm? Where have you seen Negri bodies? Of course, we know rabies shows you Negri bodies. You can have a look at this picture over here. So if they're talking about Negri bodies and you say that how will I identify this in the paper, because all of them look like round bodies. So they will always give you a history of an animal bite, dog bite, cat bite, कुछ animal bite का history होगा. which has to be there for you to think of Negri bodies for rabies. Of course, that's going to be there. And please note the site for this is going to be the cerebellum and the hippocampus. The site in which the Negri bodies are seen is going to be the cerebellum and the hippocampus. Coming all the way back, Negri bodies are done. We have Paston and Guarneri. So remember, one is seen in variola and one is seen in vaccinia. There's a lot of confusion amidst it. I hope I'll be able to sort it out. Remember, when I'm talking about vaccinia, vaccinia is looking like a G. So remember, so C may say you have made a G out of it. So vaccinia is seen in, vaccinia shows you Guarneri bodies and variola shows you Paston bodies. These are all bodies. So Negri bodies are seen in rabies, Paston bodies are seen in variola, Guarneri bodies are seen in vaccinia. There's another mnemonic for that if you get confused between variola, vaccinia, remember chota wala bodies, chota wala naam, bigger bodies, bigger name. Okay, but I think I prefer vaccinia looks like a G, so that is going to show you Guarneri bodies. Okay, where do you see Bollinger bodies? Whenever you play any football, basketball, तो वहाँ foul होता है. Bollinger bodies are seen with foul pox. Bollinger bodies are seen with foul pox. Now, where do you see molluscum bodies? You will say, ma'am, really? Are they going to ask me a question? Where do I see molluscum bodies? Of course, molluscum contagiosum. So, what is the name of these bodies? The name of these bodies is the HP bodies. And of course, I see it like this in molluscum contagiosum. So, a quick recap of the questions that you get. If you're talking about molluscum contagiosum bodies, actually what bodies are you talking about? You are dealing with the HP bodies. First, you guys will have to tell me the full form of HP bodies. HP bodies stands for Henderson-Patterson bodies. HP bodies are Henderson-Patterson bodies. And which condition am I saying it that you see it in? You see it in molluscum contagiosum. Why am I writing it again? Because there is in ophthalmology another HP body, and the name of that HP body is Halber-Steeder-Prowaski body. It is Halber-Steeder-Prowaski body. So where do I see Halber-Steeder-Prowaski body in ophthalmology? It is seen in trachoma. It is seen in trachoma. So please note, both of them are written as HP, but the word is different. One is trachoma, bacteria, the eye wala problem. One is molluscum contagiosum, virus, skin wala problem. Totally different. So here what I'm talking about is HP bodies is gonna be the very, very famous Henderson-Patterson bodies. Ab kaisa dikhta hai? So firstly, you can see that molluscum has resulted in a cup-shaped lesion. Can you see this? So molluscum contagiosum results in a cup-shaped lesion, and if I zoom into this cup, I can definitely see the beautiful pink color bodies from all over. The beautiful pink color bodies are noted. So please note, this is referred to as the famous HP bodies. I will be teaching you a lot about it when we go to molluscum contagiosum. ठीक है, तो यहाँ तक हो गया. All of these were in the cytoplasm. All of these were in the cytoplasm. So repeating, Negri bodies for rabies. Let's try and hide it and let's see if we are able to understand this by it. So if I hide all these, let's see if you are able to answer it now. Where do you see the Negri bodies? Negri bodies are seen in rabies. Okay. Next, Paston bodies and Guarneri. The Guarneri mein vaccinia and Paston becomes variola. Bollinger, ball, foul pox. Molluscum, obviously molluscum contagiosum, HP bodies. So we have done all of these. All of these were in the cytoplasm. Now I have to go on to the nucleus. So in nucleus, you please remember there is a mnemonic which students learn as Hey Bap. Okay. Hey Bap is the mnemonic. So firstly, you should know that in the nucleus, you have two types of bodies called Cowdry A and Cowdry B. So first A for you can learn as Cowdry A ki baat hogi, and B say you can learn that in this they are talking about Cowdry B bodies. Now you will know what are the viruses that I'm talking about. So over here the Y and the H. So let's begin. The Y is going to be yellow fever, and yellow fever shows you which bodies? Tories bodies. Dekho double. Yellow fever is going to show you the presence of Tory body. Ho gaya? Yellow Tory. Perfect. Let's move further. The Y is done. What is herpes? H for herpes. Herpes shows you a very famous body called the Leib-Schultz bodies. It shows you the Leib-Schultz body. So repeating once again, the H is for herpes, which shows you Leib-Schultz bodies, and Y is for yellow for Tories bodies. What is Hey Bap? B for Cowdry B, and these are not any names, but you just need to know that adenovirus and poliovirus. Adenovirus and poliovirus can show you the Cowdry B bodies. So Hey Bap is clear with everyone. Out of these, I want to show you this body as the presence of a symbol, because this can come in the paper. So this is the typical effect, cytopathic effect that you see with herpes simplex virus. The herpes simplex virus ka 3M is what is very, very famous. Please note, what does herpes simplex virus show you? Are these cells एक nucleus लग रहा है या बहुत सारा? It's looking like a lot of nuclei. So it shows you multinucleation. Next, not only are there multiple nuclei, they're all fitting into each other like pieces of clay. They are molding into each other. So there is molding. So look at this. There's 1, 2, 3, 4, 5 multinucleation. All of them are trying to take each other's shape. So there is molding. And in some cases, you see margination of the chromatin. So not so over here, but in many of the cases, you will see that they are more dark at the periphery. All the chromatin has undergone margination. So if you see this 3M phenomena, and which bodies, I just told you, hey Vap, where does it enter? It enters the cow dry type A. Yes, it shows you cow dry A. These are the cytopathic effects of herpes simplex virus. So remember, herpes simplex virus, which shows you the cow dry A bodies, are going to also show you the 3M phenomena. So till now I said either the bodies were in the cytoplasm or the nucleus, but is there any condition where the bodies are seen both in the cytoplasm and the nucleus? Yes, there are two conditions. One is cytomegalovirus and one is going to be the measles virus. I'll give you the diagram first. See, cytomegalovirus, I think by now, even without reading virology, all of you know that cytomegalovirus shows you the very, very famous owl eye inclusions, है ना? So you all have heard कि अच्छा intranuclear owl eye तो पता है ma'am, but today I'm telling you that it also shows you intracytoplasmic inclusions. It shows you intranuclear and intracytoplasmic inclusions. Then what have you heard about measles? About measles, you have heard this very famous giant cell. सोचो कौन सा giant cell in pathology have you read? M का opposite I used to tell you. M का opposite W. So we used to read the very famous Warthin-Finkelday giant cell, है ना? And Warthin-Finkelday giant cell also, if you guys remember, I had drawn in pathology, I had drawn that it had intranuclear inclusions. And I had told you it has intracytoplasmic inclusions also. So intracytoplasmic and intranuclear. Do you want to see the pictures? CMV and measles dono ka. So have a look at this. Can you see, guys, the famous owl eye appearance? Can you see there is an intranuclear inclusion? Can you see there is an intracytoplasmic inclusion? Yes. Let's talk about the measles. Measles में क्या है? The M का opposite W, Warthin-Finkelday. Okay. Can you see that there are dots inside the nucleus and the cytoplasm is also very, very pink because there are dots inside the cytoplasm also. So there are dots inside the nucleus, dots inside the cytoplasm, again intranuclear and intracytoplasmic. Just two you have to know, cytomegalovirus and measles virus. Finally, the cytopathic effects have come to an end. Now there's a short topic called assays. Assays matlab, how do I know whether this virus is infective or not? There are some physical methods, there are some chemical methods. Physical methods में you will say, ma'am, either you can do a polymerase chain reaction like COVID के लिए कर रहे थे, for RNA viruses, DNA viruses, you do that. Or right now you showed me so many microscopic photos, you look at it under a microscope. So physically, if you want to see the virus, you do a microscopy test or you do a polymerase chain reaction. But if you want to do some chemical methods, दो टाइप केमिकल में दो टाइप होता है सी यू मस्ट हैव सीन टू टाइप्स ऑफ रिपोर्ट्स फ्रॉम अ पैथ एंड अ माइक्रो लैब वन वे दे से पॉजिटिव रिपोर्ट नेगेटिव रिपोर्ट मतलब दे विल से एंटीबॉडी सीन और नॉट सीन इन द सेकेंड टाइप ऑफ रिपोर्ट्स दे से एंटीबॉडी सीन एंड द वैल्यू इज 1000 तो एक क्या है प्लस माइनस रिपोर्ट प्रेजेंट एब्सेंट क्वालिटेटिव रिपोर्ट वन इज क्वांटिटी दे आर टेलिंग यू कि एंटीबॉडीज आर 1000 एंटीबॉडीज आर 500. Similarly, for viruses also, there are two types of tests. One which tells you virus is there or not, means that is going to be the qualitative test. Endpoint assays. Endpoint assays mean end bata diya. Virus is present or virus is not present. Bas. But I say, what are the quantitative tests which tell me how much of virus is present? And that is plaque and POC assays. So please remember, they ask you in a question. In the previous year, they have asked you, which is the methodology which helps me in knowing the quantification of the virus? And that is plaque and POC. Plaque and POC are going to count it for me. Endpoint to kya hota hai? Endpoint just tells me virus hai ya nahi. So simple. Physical methods, PCR, electron microscopy. Chemical methods, qualitative and quantitative. This was one short topic. The last topic that we have in general virology is interferons. I have nothing to teach you because everything in patho I have finished. It's a repetition. Let's begin. What are interferons? Interferons are cytokines. Cytokines that are produced by, that's the question that has come back to back many years. Who makes these cytokines? We make the cytokine. I am the one who's making it, host. So if they ask you, are cytokines virus specific or are they host specific? So cytokines are going to be host specific. Every year, see, once upon a time when there was a PGI ka exam before INICT, PGI used to give this in every paper. Interferons was their sought after, hot trending topic. So now you expect it in future exams. So this is host specific. It is made by the host. So how many types of interferons are you aware of? We have interferon alpha, we have interferon beta, and we have interferon gamma. So interferon alpha, beta, and gamma are the ones that we have. So your question first to me will be, ma'am, दो questions पढ़े थे when we did pathology. Where do they come from? So question number 1, what is their source? And question number 2 is, what is their function? So please note, the source was LFT. What is LFT? L is for leukocytes. Which leukocytes? Very commonly it comes from macrophages. Okay, F is for fibroblast and T is for T helper 1 cell. So repeating, what is the source of interferon alpha, beta, gamma? LFT, leukocyte, fibroblast, T helper 1 cells. Then what do they do? What is their function? So alpha, beta, gamma का function है alpha, beta and gamma. How easy? Alpha, beta, gamma का function alpha, beta, gamma. So A means that this has antiviral properties. It has antiviral properties. That is why now they are saying that let's use it for making some antiviral drugs also. For that matter, even B has antiviral properties. So you can say interferon alpha as well as interferon beta, both of them have antiviral properties. But primarily A for A. Beta का अलग वाला function क्या है? Beta I will learn for brain. What is it used for in medicine? It is used for the treatment of a brain disorder, that is multiple sclerosis. So interferon beta has been used for the treatment of a B for brain disorder, multiple sclerosis. And what is G? G for granuloma. Guys, this is important for two things. Number 1, interferon gamma is needed for granuloma formation. And number 2, is it used in the treatment of any disease? Chronic granulomatous disease. Padha hai na pathology and medicine mein. So, number 1, interferon gamma is needed for granuloma formation. And number 2, it is needed for the treatment of chronic granulomatous disease. Ho gaya khatam. Interferon alpha, A for A, antiviral property. Interferon beta, beta for brain multiple sclerosis treatment. Interferon gamma, G for gamma, granuloma formation and granulomatous disease ka treatment. Now, if you say that, can you tell me that this is something that I'm telling you only from the point of view of INICT? So, if you ask me the chromosomes, so you will say that, ma'am, for the formation of interferon alpha, interferon beta and interferon gamma, if I ask you, what are the chromosomes that are involved? The chromosomes are going to be 9, 9, 12. These are the chromosomes for the interferons. But if you say that, ma'am, all these interferons will go and act on some receptor also. So what are the chromosomes for interferon receptors on which they go and act? And that is 21, 21 and 6. कैसे याद करें? So let's read it. Rather than learning it like this, I will say pick up 1-1 combination. Interferon alpha comes from which chromosome? Acts on the receptor, that comes from which chromosome? So everywhere you're seeing 921, 921 का symbol बन रहा है. ठीक है? Interferon alpha comes from chromosome 9. Its receptor comes from 21. Beta also comes from chromosome 9 and its receptor comes from 21. तो पहले तो 921, 921 याद कर लो. And then you do an inversion. Not only in the cross connection of the size, also flip the numbers. मतलब 21 को flip करा तो for interferon gamma, what have you got? 12. And 9 को flip करा तो you get a 6. So simple. If you flip the 9 like this, you are going to get a 6. And if you flip the 21, you are going to invert it, you will get a 12. So that is the best way you could learn it. So for interferons and for their receptors, alpha ke liye 921, beta ke liye 921, flip kara, that becomes 12 and 6. That is something which is important. Again, this was once upon a time a PGI favorite and that is the reason that we've got it. But what is most important going forward? Number 1, you should know LFT and ABG. Wo yaad ho gaya. And remember, these are host specific. We are the ones who have made it. That marks the end of our general virology, and now we are going to begin our journey with individual viruses. But now life will be very simple because you have learned all the general